Vibrio and like organisms, or BALOs for short, are a group of predatory bacteria that prey on gram-negative bacteria. These organisms are highly motile and can be said to be important bioagents in controlling bacterial populations. Environment and ecology. BALOs are bacteria that can be found in many different environments, some of which include saltwater, freshwater, soils, and even animal bodies. Temperature, prey, and salinity, which is the concentration of salts in soils and water, are very important in determining the growth of BALOs. These factors can impact the distribution of BALOs' habitats, their diversity, as well as prey determination. More information can be found under the Environment and Ecology tab of the website.
Taxonomy. Taxonomy involves naming, identifying, and recognizing organisms based on shared characteristics. Taxonomy is so important as it allows us to categorize and study organisms in the field or in the lab. It also lets us communicate about our research, as the organism can be universally recognized under the same name. Vibrio, for example, can be identified by its shape. It is a comma-shaped motile rod with a single flagella. It is gram-negative, highly motile, mesophilic, and parasitic. Many biochemical tests can be run to identify the specific genus and species of the organism. Vibrio is classified under the bacteria domain, Vibrio genus, and has many subspecies within its classification. A phylogenetic tree diagram presenting the different species within its class can be identified on the website under the taxonomy section.
General phenotype. Whereas the genotype of an organism refers to its complete genetic makeup and the complete description of the cell's genetic information, the phenotype relates to its observable characteristics. Vibrio is an example member of the BALOs phenotypic group, and their life cycle is complex and multi-stage. Throughout the cycle of predation, which begins and ends with the release of progeny cells, the surface structures of the predatory bacteria and the enzymatic gene products are of great importance to the continuation of the cycle of predation. Two surface structures that are important to the predatory life cycle of most BALOs are the flagellum and the type 4 pili. The flagellum allows the bacteria to move through its environment and therefore collide with prey bacterial cells. The motile period of the predation cycle is referred to as the attack phase. Type 4 pili play an important role both in the initial adherence of the predatory bacterium to its prey and the entry of the predator into the prey periplasm. Glycanase enzymes are important for the generation of the penetration pore that allows the predatory bacterium to enter into the prey periplasm. Hydrolytic enzymes are important across the life cycle of the predatory bacterium as a whole, from aiding the generation of the penetration pore to metabolizing the solutes extracted from the prey cytoplasm and controlling the lysis event that allows the release of the progeny cells. More information relating to this subject can be found under the General Phenotype tab of the website.
Selection pressures and unique phenotypes. A selection pressure can be defined as a change in the external environment that drives phenotypic change. Vibrios live in a very variable environment, so there are many different selection pressures that change the way the phenotype is expressed, depending on the strain. Some selection pressures include the availability of amino acids in the environment, competition with other BALOs and availability of prey, and unfavourable conditions that are liable to change, such as salinity, temperature, or lack of nutrients. On the screen are some examples of phenotypic responses. One example of phenotypic change in response to unfavourable external conditions is the ability of some BALO species to arrest their life cycle at the stage of delaplasts formation. Delaplasts are stable, spore-like structures that protect the BALO species. The structure reduces rate of respiration, resists external changes affecting internal structures, and reduces the need of ATP, the amount necessary for survival. This allows for the BALOs to exist in dormant form for a long period of time. Once conditions become more favourable, the life cycle will continue. More information can be found under the Selection Pressure tab of the website.
Example organisms. Until recently, Delovibrio bacteriovorus were the only recognized species of BALOs. However, recent research have uncovered many other species. For example, Delovibrio exovorus and Bacteriovorax marinus. If you want to know more about these specific species of BALOs, they can be found here under the example organisms page on our website.